First, I thank you very much for the introduction. I thank also the organizer for inviting me, giving me a difficult task. One is talking about what is increasingly recognized as being a central role of the GI tract in CNS disease, and in particular in neurodegenerative disorders. And although we are aware that there are increasing data showing links between dysfunction of the gut, dysfunction of the NS, and CNS disease, there's still it's still an emerging concept, and I think there is, although prodromal disease for which this link exists, and this is Parkinson's disease, and I will focus my talk on the link that could exist between the gut and the brain in Parkinson's disease. And of course, the second hardest task I have is talking about Michael after his brilliant talk, so I think I will try to keep up on what you did. The organization of my talk will be done along three major axes. The first one is try to define you and to go a little bit into the insights of the pathophysiological mechanism of Parkinson's disease, and trying to define you, giving you insight into its mechanism as being a prion-like disease. Second, I will try to prove you evidences that are gained both from animal models but also from patients, suggesting that, at least in part, some Parkinson's disease subtypes could have a digestive origin. And the second is try to give you also a review of the literature showing that the enteric nervous system can be affected, and in which sense this could help us either in defining early biomarker of the disease, or targets or therapeutical targets for the disease. As you know, Parkinson's disease is movement disorders. It's, after Alzheimer's disease, the second neurodegenerative disorders, and affects 1 to 2% of the population aged over 65 years. It's also a chronic disease for which the causal mechanisms are still unknown, but probably part of it, and the minor part of it, are of genetic origin, with mutation in synuclein, LAC2, PARC2, but also over 90% of the causal factor of Parkinson are probably environmental factor or still unknown genes. And among environmental factors that have been supposed or suggested to contribute to Parkinson's disease development are pesticide, viruses, or head injury, and I will come back to all these factors later on in the talk. As you know, from a clinical point of view, Parkinson's disease is characterized by three cardinal motor symptoms. That is resting tremors, slowness of movement, and muscle rigidity. From a neuropathological point of view, there are two major hallmarks in Parkinson's disease. One is the loss of dopaminergic neuron in the substantia nigra innervating the striatum, which is probably considered as the causal mechanism for the movement disorders. And second is the presence in these neurons of Lewy body or Lewy neurite, which are hallmarks, basically, of Parkinson's disease. So this makes us also go to the definition, the molecular definition of what are Lewy body and Lewy neurites. These are, of course, as mentioned, intraneural inclusion, which are either round or that can be filamentous. And the main constituents of Lewy body is synuclein, is a protein named synuclein, which is found in the Lewy body as either ubiquitinated, but also most importantly and used as a neuropathological hallmark of the disease, is in a state of a phosphorylated state, mainly in serine 129. Besides being a marker, a neuroanatomopathological marker of the disease, alpha-synuclein has also a functional role. And as you can see here, or you should see here, if the picture would be more magnified, it's found in green here in presynaptic terminals. Here you see a MAP2 staining of dendrite, and you see a strong concentration of alpha-synuclein in presynaptic terminals. And its role has been, its physiological role has been the most well described, although only partially, as an actor of synaptic transmission, because it participates in the filling of synaptic vesicle, in the formation of this vesicle, and the release of the neurotransmitter docking to the membrane and release of the neurotransmitter. This is the physiological role that is still being investigated. However, besides its physiological role, alpha-synuclein has also a pathological role and a role that is considered as a prion-like protein because it's in its normal physiological condition, alpha-synuclein is found as a soluble form of monomers in dimers that are either in the cytoplasm or membrane-bound. However, in presence of alpha-synuclein seed, for which the seeding process is still unknown, this alpha-synuclein starts to oligomerize and forms fibrils that are considered as toxic for the nervous system, as you will see. And after this fibrillation processes, you have still a continuous oligomerization that gives you amyloid aggregates that you found, for instance, in the form of Lewy body or Lewy neurites. And the toxicity of these amyloid aggregates is probably relatively low. The toxicity occurs here. And this is just an electron microscopy of these fibrils that you can see here, how they form after you put monomer and seeds. You can see after 5 hours' time, it's a dynamic process. You can see the formation of these fibrils. And then this is schematic what occurs in a neuron and what allows for the transmission of the disease. This is the seeding process, how from a normal physiological protein you can get toxic oligomers. And this explains you how the transmission can occur because these fibrils have the capacity of leaving the neurons, either when the neuron dies or with a dynamic process of exosome secretion and being uptaken by second neurons and being seeded further. And then transmission being a way to carry the disease from a neuron to the other neurons. And what I will now focus just with some examples is to describe you the toxicity of these fibrils and that you need this dynamic process of formation of fibrils to express toxicity. These experiments that have been performed where different forms of monomer and fibrils have been added to neuroblastoma cell line, and they've looked the impact of this addition upon the survival of or upon apoptosis. And what you see is that if you add monomer alone, you don't have any apoptosis in this neuroblastoma cell line. If you add fibril alone, you don't have any apoptosis. But if you add fibril and monomer, you have the induction of apoptosis, suggesting a dynamic process. And this was further highlighted both from a mechanistical point, what occurs at the cellular level, the cell membrane level, when you have the formation of extracellular fibril. You can see here, when you add the fibril alone, you see over time, 1 day, 3 days, and 4 days, you have a loss of membrane integrity, of progressive loss. However, when you add in hippocampal neurons, this time, fibril and monomer, you can see a significant increase in the speed and in the intensity of the membrane damage. And this dynamic process of toxicity is, of course, inhibited if you add a blocker that prevents the formation of the fibrils. So based on this physiopathological mechanism, meaning seeding and transmission from one neuron to another neuron of a toxic protein, you can now apply these physiopathological processes to the topographic expansion of Parkinson's disease in the brain. And what has been shown, and this is work done by Topographic, is that he characterized the lesions of the brain in Parkinson's disease patients as a function of the severity of the disease. And the stage 1 is the less severe disease with premotor symptoms. Here you have motor symptoms, and there you have the addition of other symptoms which are non-motor, non-dopaminergic symptoms, such as dementia or other dysfunction. And what you see in all patients is you see that in the least severe, there is always a common brain lesions, that is the dorsal motor nucleus of the vagus, that is affected, in which the presence of aggregated alpha-synuclein has been observed independent of staging. And after time, you can see a coderostral progression of the lesions over time and over severity along the ceruleus, along the substantia nigra, and the mesocortex, which progressively invades other structures of the cortex. So you have, in this stage, a progression of the disease from a region which initiates in the dorsal motor nucleus of the vagus and then affects in a caudorostral adder region, suggesting that the origin of the disease could lie even beneath the dorsal motor vagus, the dorsal nucleus of motor vagus, sorry, which is what he supposed that its origin could be in the enteric nervous system, and based on the progression of the disease, you could have an entry route from the gut towards the brain. And this is where I will try to focus now, trying to demonstrate what are the evidences that show that indeed, when you inject or apply toxic forms of alpha-synuclein in the gut, you can have a rostrocaudal transmission along the vagus nerve. And this has been shown very recently in some experiments where they injected in the duodenum at multiple sites either lysate of brain from Parkinson's disease patient or synuclein fibrils, and they characterized over time the evolution in different brain structure. And what they showed after 12 hours of injection in the muscle layers, here they injected, for instance, BSA albumin as a control. They showed that after injection 12 hours, it was localized either BSA, lysate, phosphorylated alpha. They used an antibody against synuclein to identify this deposit of synuclein, and they found it in the duodenum 12 hours injection. And what they saw after is that rapid progression after 2 days along the vagus nerves, and after 6 days, they could reach the brainstem region, suggesting that indeed there could be a transport and a progression of alpha-synuclein along the vagus nerves to reach the brainstem. Conversely, however, another group also showed that this was not a unidirectional propagation, but you could have the same region by injection of this time viral surexpression of human alpha-synuclein that has been shown to induce, by surexpressing alpha-synuclein level in neurons, you can have the formation of aggregates. And what they have shown, they have looked when you injected human alpha-synuclein in the rostral brain, you could have a propagation from brain structures toward the dorsal motor nucleus. And after they looked, after some time, whether such propagation could also be found within the stomach. Indeed, they could see after 6 months injection, you could have some presence of aggregate of alpha-synuclein, phosphorylated alpha-synuclein in the stomach, in fibers of the stomach plexus, probably of vagal efferent origin. And this was amplified after 12 months, suggesting that indeed there could be some propagation. However, experiments where they tried to do vagotomy was not performed to formally prove that the vagus nerve was indeed conveying alpha-synuclein either from the gut to the brain or to the brain, from the brain to the gut. But there are evidences showing that such propagation could occur from data obtained in patients. Indeed, this is recent from cohort patient, from the Sweden and Danish cohort patient, where they compared the cumulative risk of developing Parkinson's disease in patients having been vagotomized in the 70s, mainly for treatment of peptidic ulcer. And so what they looked is the cumulative risk from patients here in a cohort patient. They can show that you have a cumulative risk over years of developing Parkinson's patient that is not affected when you do supraselective vagotomy. That means that you just cut the innervation probably of the upper part of the stomach. However, when you do truncal vagotomy, there is a significant reduction in the risk of developing Parkinson's disease. Alternatively, these results were already confirmed in another cohort, another Swedish cohort that was just published, where again, selective vagotomy did not prevent the risk of developing Parkinson, while truncal vagotomy was able to reduce significantly the risk. However, as you see, the risk was not reduced to zero, suggesting that there could be still other entry roads or other mechanisms for Parkinson's disease. And other entry roads of Parkinson's disease have been suggesting, for instance, via the olfactory bulb, in terms, for instance, of infection of the splenic fluid. There was a high surge of Parkinson's disease, sporadic Parkinson's disease. And this has been confirmed that indeed the olfactory bulb can be an entry site for Parkinson's disease, where they have injected in an experiment, they have injected synuclein fibrils in the olfactory bulb and looked at the progression over different brain regions over time. And indeed, they have shown in this graph where you see the brain structure, different brain structure, and the evolution of the different colors represents the sites in the different brain regions where you have the presence of phosphorylated alpha-synuclein after injection of the recombinant fibrils. And you can see that you have a progression from the olfactory bulb to different regions, again in a caudorostral direction, with a deeper brain region being affected, such as the locus ceruleus or the substantia nigra, after 12 months have been injected. However, these animals, although they exhibited motor smell deficits, they did not exhibit motor function. So this is still something to go further on. So now the question is, if we know now that pathological forms of alpha-synuclein could propagate from the gut to the brain, what can be the causes? What causes in the gut this formation of aggregation of alpha-synuclein? And one nice experiment done by Paco Panmontorio have shown that injection of drugs such as pesticides, such as rotenone, which is an inhibitor of the respiratory chain of the mitochondria, could reproduce neuropathological symptoms of Parkinson's disease. Indeed, I apologize here, you don't see very much the color, but what you can see is the staining of the myenteric plexus after 3 months chronic injection of rotenone. And you can see here normally stained alpha-synuclein in control animals. However, you can see the presence of aggregated alpha-synuclein 3 months in the myenteric plexus. And this is also observed in the brain, in the dorsomotor nucleus of the vagus, where you have a significant increase in alpha-synuclein immunoactivity after 3 months of injection of rotenone in these animals. And what they have shown here, that if you vagotomize the animal, you are able basically to prevent here the expression of alpha-synuclein, the increase in alpha-synuclein in the dorsomotor nucleus of the vagus, suggesting that indeed there could be a propagating route, further suggesting that this propagating route exists and that rotenone can induce this pattern. Another actor that we heard a lot about at this meeting that has been suggested also to be involved in Parkinson's disease is the gut microbiota. And experiments have been performed in a model, a transgenic model of mice surexpressing the human form of alpha-synuclein, which is a good model of Parkinson's disorder, which presents pre-digestive prodromal symptoms and motor deficits, as well as neuropathological lesions of alpha-synuclein. And what they have studied in this mice model of Parkinson's disease, the impact of the presence or absence of microbiota upon motor and GI function, as well as alpha-synuclein pathology, and how by manipulating the microbiota you can affect these processes. And basically, to make the story short, what they have shown is that in germ-free animals, that motor deficits have been significantly reduced as compared to control animals, and as well that the alteration of GI transit, and in particular the reduction of the fecal pellet output observed in transgenic animals, in Parkinson model animals, is reduced in germ-free animal. And similarly, just to make the story short, is that the neuropathological lesions of alpha-synuclein, that is the presence of insoluble forms of aggregated forms of alpha-synuclein, is absent in the substantia nigra of germ-free animals as compared to SPF-free animals. Not the total synuclein, but only its aggregated forms. What are the factors here that could contribute to this dysfunction of alpha-synuclein is still unknown. They have an hypothesis, which is a little bit, we heard a little bit about short-chain fatty acid, how good it is, but here they have shown that indeed short-chain fatty acid, and in fact butyrate is a metabolite produced by this bacteria that enhances the development of Parkinson's disease. So they suggest that then when they administered high doses of butyrate, which is increased in this model, you favor the development of Parkinson's disease. So this is again a question of cause of maybe of dosage of butyrate concentration. However, there could be other mechanisms which I can go over here, this is just it's important, that could contribute, for instance, to the pathology of Parkinson's disease is the ability of the bacteria by themselves to be producer of functional amyloids that have been named, that are named as curly, that are produced naturally by bacteria when they colonize the gut to serve as a biofilm, and they use also as a quorum sensing during this competition in the colonization of the gut. And it is known that amyloid can cross-seed each other, suggesting, for instance, that curly can induce aggregation of alpha-synuclein. And to test this hypothesis, they have injected rats, aged rats animal, which have already a proportion with aging to increase, to present phosphorylated aggregates of alpha-synuclein. And in this rat animal, they have injected them, they have gavaged them with bacteria, which are E. coli strain, that produces this curly form of amyloid protein, and they have looked at the capacity of amyloidogenic proteins of bacterial origin to induce seeding and aggregation of alpha-synuclein in the gut and in the brain. And indeed, this is surprising, what they have shown is that when they look at the presence of phosphorylated aggregates in animals as compared to controls that have been gavaged that produce this curly protein, you have a significant increase in aggregation of phosphorylated alpha-synuclein, both in the gut and also in the brain as compared to when they have gavaged with the strains with an operon mutant for the production of this curly protein, suggesting that the process of protein aggregation could start in the gut and be propagated towards, start within the gut and be propagated within the host, in the intestine, and then toward the brain. And so, and this is what I will show after, is still speculative. So what could be an intermediary between the microbiota and the host to favor this propagation? And there is a cell that has been recently shown that we know all of, that we have all heard, that is enteroendocrine cells that are able to sense the luminal content and then transmit it to the host. And what has been shown is that this enteroendocrine cell by the group of Lidl these neuroendocrine cells contain or express highly alpha-synuclein. So this could be a way how amyloid protein in the gut could seed aggregates via enteroendocrine cells and then via the enteric nervous system toward the brain. I apologize for the poor quality here. And what you can see here, for instance, this is done in mouse where GFP mice express PYY expressing cells, PYY expressing GFP cells. And here in human colon, you can see also that indeed PYY cells, enteroendocrine cells, co-express alpha-synuclein. So this is a way how you could have a route, basically from the gut to the brain. So now the question that I will just tackle in the few minutes left is whether the enteric nervous system is also affected in Parkinson's disease, because we know that Parkinson's disease patients have GI dysfunction that appear probably many years prior to the development of the motor symptoms. And ENS lesions have been observed, of course, since the early 84 in the lower esophagus, in mesenteric plexus. This has been further confirmed later on in autopsy patient in 87 and in 88 by Wakabayashi, showing that the whole digestive tract could be affected with a rostrocaudal density of lesion being higher in the upper GI tract than in the lower GI tract. And then we have also further, this has been an autopsy patient, and we have also further contributed to show that in living patients we were able to routinely to detect the presence of aggregated alpha-synuclein, phosphorylated alpha-synuclein in the enteric nervous system, with Lewy neurites mainly in the submucosal being present in 72% of PD patients and Lewy neurites being absent in the control. And what is interesting, this could be used as a differential marker for Parkinsonism syndrome, because in other Parkinsonism syndromes, such as multiple system atrophy or progressive supranuclear palsy, there was an absence of aggregated alpha-synuclein within, or phosphorylated alpha-synuclein within the enteric nervous system. And of course, not all patients, as mentioned here, have also the presence of phosphorylated alpha-synuclein pathology in the gut. Then what is also interesting is that there was a good correlation between the severity of the non-motor symptoms and the burden of the neuropathological burden of the lesions in the ENS. When we graded the number of lesions of alpha-synuclein present in biopsy, we could show that there was a good correlation between the Axel score and this severity burden. And what is also interesting from other studies from the group of Shannon and Cordova is that this could be used at looking at the presence of phosphorylated alpha-synuclein in gut biopsies as a premotor biomarker of Parkinson's disease patient, because what they have shown in this case report studies, and this has been confirmed in a more recent study, is that they have gained access to biopsy from patients that have been diagnosed years later with Parkinson's disease, and they were able to go back to their anapathological, to biopsies that were taken prior for cancer surgery. So when they looked for the presence of aggregated alpha-synuclein in this patient that at the time did not have Parkinson syndrome, they were able to identify the occurrence of alpha-synuclein staining prior to the development of the disease, further supporting the hypothesis that this could be used as a biomarker of premotor biomarker of the disease. Another issue that has been long debated, but that is, I think, getting clear, is whether Parkinson's disease was associated with a loss of enteric neurons in the enteric nervous system, and neither in the myenteric plexus nor in the submucosal plexus there is a significant loss in myenteric neuron. And the only morphological changes, when we talk about morphological changes that have been observed, is that there is a significant reduction in ganglia volume in the enteric nervous system and a significant increase in the number of mitochondrial cells, without knowing what is the functional impact upon the enteric nervous system. So there is another recent study from the group of Roberto Di Giorgio that further identifies, however, neuroplastic changes in the enteric nervous system. They confirm first that there is no major neuronal cell loss. However, there is a significant reduction in the proportion of viapirogic neuron in the enteric nervous system, and suggesting that this could contribute to some dysfunction. And finally, one of the major changes that has been reported in the gut recently in patients with Parkinson's disease is a significant loss of the acetylcholinesterase density, especially in the upper part of the GI tract using PET tracer. This is in control group. You can see the intensity of acetylcholinesterase density, and this is in Parkinson's disease patients. And the significant decrease, whether it's attributed to the enteric nervous system or contributes to just to vagal degeneration, still remains to be defined, but this could contribute in part to GI dysfunction observed in Parkinson's disease. So to conclude, the gut is probably a central actor in the physiopathology and etiology of Parkinson's disease. Probably it's a very complex disease, but the enteric nervous system could be a source of biomarkers for Parkinson's disease, especially in pre-symptomatic stages. And also targeting the gut could be a therapeutical target. And the question arises whether this propagation from the gut to the brain could be observed in Parkinson's disease is also true or not for other neurodegenerative diseases, remains to be identified. So I want to thank all the people of the group, of the lab, and especially much of the work that I presented here was done by the Pascal Berkinder from our group. So I thank you for your attention, and thank you.